Okay, I'd like to welcome you to this session. We just had a great session on CAR T cells here at the iwNHL meeting, and my colleagues we had a very spirited discussion. So why don't we dive into some of the things we found? So one of the issues was we have 3 products, 2 approved, 1 almost approved, hopefully, in diffuse large B-cell lymphoma. What is your impression of the data?
I mean, I'm delighted to see that we have 3 products that are effective and durably effective. I think it supports, this therapeutic approach as what should be a standard, at least for the patients now that have failed 2 prior therapies with aggressive lymphomas.
What was the most impressive about the data?
I think the durability is the most impressive thing. We have multiple trials now that show very durable responses with remissions lasting for years, and including remissions that continue after normal B cells recover. So patients don't really have to worry about long-term lack of immune responses, which was one of the things we were majorly concerned about before.
You know, most of the patients were immunodeficient going into the trials. They had low antibody levels, 0 B cells from prior Rituxan, and many of them had marginal even T cell counts from past therapies. Once the disease is eradicated and the patient is in remission, you begin to see all these parameters improve. The immune system improves in the absence of disease, and as well as performance status, cognitive function, and everything. So being in remission is clearly the best way to be.
And as a transplanter, this is only 1 dose. This is 1 dose. Nothing was done in consolidation. Nothing was done in subsequent therapy. And the B-cell recovery and then the amounts of long-term safety is what I think is most impressive compared to other things that you're seeing with not just even autologous stem cell transplant or even allogeneic stem cell transplant. One thing I think unique across all these trials is that these patients could never even get to a transplant. And this is why I think this is paradigm shifting for large B-cell lymphoma.
But all that great hype we had, and so there's great results, but there are some challenges. Not everyone has a CR, a long-lasting CR, so we're at about the 40% range. So that means about 60% of people, still either relapse or don't respond. And there are issues of toxicity. What are your thoughts on that? I mean,
yes, we're nowhere near 100%. 40% is not 100%, but I think that we're poised to begin to understand that 60%. Samples have been collected from products, from T cells during expansion, tumor biopsies to look at microenvironment. All investigators in this field now have collected samples and are in the process of analyzing them. And so I think it's really likely that we're going to figure out, and it won't be one solution for everybody, but we're going to figure out what the factors are that determine failure for the 60% that don't fail, or that aren't successfully treated. And the toxicity, we can talk about, but I think the toxicity is very manageable in lymphoma patients, but you have to be educated, as I think Bill said in his presentation.
There's a lot of room for improvement. I mean, we don't want to stop here at Kite Gilead. I mean, I agree with you that we can get better. And this is one of those rare times as a physician and scientist, we can go from bench to the bedside and bedside back to the bench. And we need to all work together, whether in industry, within academia, as well with the government resources, and where we're actually really understanding all these aspects. And you're right, it's not going to be one thing, it's going to be multiple things, from the incoming T cells, with the manufacturing, with the tumor microenvironment, and how to manage the patients, and potentially understanding using other agents to then improve these outcomes. And I think the work that Jim's been doing that actually accentuates that with the constructs. I don't know if you want to talk about that.
I think that's really exciting, because we've got differences between the 3 products, but we also have products that aren't even yet in the clinic, or that are barely in the clinic, and that seem to have even less toxicity based on the construct of the CAR.
Yeah, I think the design of new CARs is one of the main avenues of improvement for the field. We definitely are constantly designing new CARs in my lab, so we design new CARs like every month, and we're testing them all the time. So I think that, and many other labs are doing the same thing. So I think it's a field where there are a massive number of people involved. Large amounts of resources are being committed to this field because it's so incredibly promising. And I think definite improvement in CAR design and many other factors are going to improve this field rapidly.
But it's really hard to imagine then how do we go forward with clinical trials if we have 3 drugs approved in a setting. Then what do you do with your new CAR? I mean, it is a challenge to figure out how to break basically into the market from these areas.
Tougher than it used to be to get people for clinical trials, but you just have to have some compelling argument or some compelling reason
It's
for
actually a
patients
good
to want to
thing.
do
So
the trial.
40% of patients are not going to need, with refractory large B-cell lymphoma, are not going to need a clinical trial because they'll be in a durable remission. So the reduction in patients' availability for trials is good. Now we have a standard. If you make a new CAR, you have a standard CAR against a new CAR.
But the other thing is we're only treating relapsed refractory patients right now, and so that's obviously the toughest of the tough.
Yeah, so 40%, we're likely to do better.
Right,
in the end.
but obviously the design of trials going forward is to try to move this up another level to maybe where we're talking about some of the transplant trials. Does someone want to address the transplant trials?
Yeah, we have a Zuma-7 trial, which is our randomized global phase 3 in second line. So we're not randomizing against transplant. We're randomizing against second line, which is standard of care rituximab plus a cisplatin agent with the intent to go into autologous stem cell transplant. And so I think this is when other sponsors also have randomized phase 3s, and I think this is going to answer that question with earlier lines of therapy, healthier patients with healthier T cells. Could this actually be impactful and actually be even better benefit risk?
And you showed some interesting data that patients with less tumor burden potentially had greater, better outcomes with less toxicity. I think that's another way forward.
Yes, Dr. Locke at ASCO in 2018, we did analysis with quartile analysis for the lowest quartile of patients that had the least amount of tumor burden by sum of product diameter per Chesson, actually had the best overall responses and durable remissions, about 70% durable remissions at the patients with the least amount of toxicities.
Yeah, so we did a similar analysis using volumetric measurements on PET-CT scans. So rather than sum of products, we actually looked at the entire tumor volume, and tumor volume clearly relates to toxicity. The higher the tumor volume, there was statistically significantly more toxicity, particularly CRS. But efficacy was about the same in our analysis. So patients, clearly if you can start earlier before you have a disease that's a large volume, the treatment's more tolerable. But either way, the responses seem to be the same in our analysis.
But back to what you're saying, how you can improve. I mean, obviously, I know Jim and you, Steve, are working on other targets, and in the industry, we're also looking at other pipelines and other B-cell targets, because I think that by targeting other B-cell antigens and we're seeing antigen escape, that we can potentially enhance in other subsequent CARs as well.
Yeah, I mean, that's a key point, because antigen negative escape is obviously a limitation, but that you can also think that if you've got a tumor that didn't have as much mutational load potentially, that it'd be harder for it to lose CD19. So moving up might actually even help, but ultimately we probably need another target, right? So CD20 or another CD22 or another target where we have dual CARs or logic
just
CARs.
a mixture of
more than one CAR to make it go forward.
I think that's definitely the way the field is going. There'll probably be very few monospecific CARs a couple years from now.
And that leads to production issues, and everyone recognizes that right now it is a challenge, because you have to first get insurance clearance for the patient, then you have to get the cells collected by apheresis, then there's a period of time, a couple weeks to 4 weeks, to get the cells manufactured, and you have to keep the patient going during this period of time. And this leads to some patient selection in terms of people to be able to make it through this whole process, and that is a challenge as well. And moving it up into less disease burden might make it easier.
Absolutely.
I think that's a really important unmet need, is a really successful bridge to CAR therapy that cytoreduces patients and yet doesn't make them so sick that they lose performance status or have toxicities that prevent subsequent CAR T-cell therapy.
Seems like we still have also work to do regarding CRS and neurologic toxicity, which are the two major complications. We had some great talks and discussions about some of the insights of maybe breakdown of the blood-brain barrier or infiltration of inflammatory cells into the CNS or cytokines that are causing this. But clearly that's another way. We need a long ways to go unless we can make a safer CAR that doesn't cause any cytokine release or neurotox.
Yeah, we have a phase 1 trial where we have a CAR. It still causes CRS, but the neurotoxicity rate was significantly less than at least some previous CAR products. So it's
And that's basically a CAR that has less cytokine production, at least in vitro. Do we know about it in vivo yet?
We don't really know about in vivo. We have some preliminary data from humans, which is very early. We probably won't want to discuss that at this time, but we'll have we'll be reporting that soon. But yeah, I think CAR design to have maybe a CAR that's not quite so powerful, that where you can still eradicate the malignancy but not cause severe toxicity is a way to go.
I think there's going to be, like what you were saying earlier, like a Goldilocks or a sweet spot effect, is that can we decouple the efficacy from the toxicity? At Kite Gilead, we're looking at early markers and early signs and looking product characteristics to really try to determine and deconvolute the specific T cells and different types of cytokine productions to then optimize the next generation of products.
Yeah, so how do you maximize efficacy while minimizing toxicity? You know, there's not any drug that we use in oncology or in medicine that doesn't have side effects or potential side effects. So I don't think you'll never get to zero, but to optimize or at least maintain there's efficacy and figuring out how to reduce toxicity is one of the challenges.
But we also have a product that proliferates and expands in the patient, and the better it proliferates and expands, probably the better it works to some degree, and that also then probably correlates to some degree with toxicity. So we need to maybe figure out the right ratios. All right, this has been excellent. Any other thoughts?
No, I think it's an exciting time in oncology.
All right, thanks very much.